So the title of this talk here is Disentangling Non-Crossover and Crossover Genotype Bioenvironment Interaction for Selection. And I'll sort of get into exactly what that means, but the main components of G by E are non-crossover and crossover. And usually what people sort of say is, oh, there's G by E. You know, we've got G by E in our data, and sort of what I'm all about is, well, what's the form of it? What does it look like? Does it cause problems, or is it just sort of there? It's not causing any re-rankings. But before I get into that, just a very quick plug. Just a very quick plug. Gregor and myself are hosting the next UCARPIA biometrics meeting here in, as I said, sunny Scotland, next year, the end of next year. So I'm sure that you'll keep seeing similar pictures and things when it gets closer to that. And I know already we're going to be roping in a bit of the lab to do a bit of this planning. As Chrissy mentioned, the work that I'm going to be presenting here was a bit of the PhD that I've just finished, and that was originally instigated back in 2018 or so. Nonetheless, it's a joint collaboration between the Highlander Lab, Bayer Crop Science. So we have Gregor, that was my main supervisor, Chris Gaynor, that used to be here and moved to Bayer, and Brian Gardunia from Bayer. I'd just like to thank Bayer, obviously, for the scholarship and as well as the data, but also note that in this particular talk, there's no data from industry in here. I'm going to be using simulated data just to avoid any issues there. Just a very quick, I guess, overview of what I'm going to touch on. I'm going to talk a little bit about multi-environment plant breeding trials, what they are, and why does it GYE cause grief within those trials. How we're going to use or leverage GYE to select for broad and specific adaptation within those. How can you do a little bit of a simulation to simulate some GYE, what that looks like, and then formally, how do we disentangle the components that I mentioned before, non-crossover and crossover. But of course, I got to mention, sort of define what those things are. And this one is a bit more plant-based, but some of the methods are very applicable to animal-based. I guess the key difference is in plant breeding, we have a large number of environments instead of sort of just a select few. But all the concepts or the theories sort of applicable across the board. So when I talk about plant breeding, I guess I've just got a little small schematic here. We've got multiple stages of testing and selection. I'm not going to harp too much on the sort of the pipeline here because it's less about that pipeline. It's more about the field testing. Nonetheless, we have an initial cross between parents. We might make 100 crosses, say. You get 100 families, biparental families from that. Within each of them, you might have 100 individuals within each biparental family. And then you take those and you select them, you put them in the field, you test them for things like yield or protein, whatever the certain traits of interest. And the key point here is that you're starting off with a large number of genotypes and you're dwindling down to something at the very end, which is eventually going to be released to a farmer in their field. That might be one single variety. So you're going from a very large cohort or population, eventually going down. In this sort of little schematic here, we've got double haploid lines so that they're fully inbred. So even though you form the F1s, you go and form a double haploid from that. So that each one of those 10,000s is, we can say it's a unique genotype. On the other side of the coin is, as you're going down, you're making your selections, you're selecting for things like protein or yield, disease traits, you're putting them in more environments. So what a breeder's doing is that it's selecting material that is well adapted for what we say is their target population of environments. And that represents, essentially might be the region of intended production. It's all the farms that are sort of in his region, his or her region rather. And that's what he's going to be targeting. So when I say, oh, they're targeting yield, they've got to be well performing in terms of yield within that population of environments. So there's a large number of environments that constitute that. And within a plant breeding program, what they're doing is they're taking the material and they're putting it in select few of those environments, because every year they're running field trials. And the field trials that sort of look like this here on the left-hand side. This one's actually been very well maintained, because usually plant breeding trials don't look that well groomed. But what's happening here is we've got a series of rows and columns, and then within each one of these plots there contains a unique genotype. And that whole sort of trial here has got a number of genotypes. So it might be 9 by 7. So what's that, 63 different genotypes. And then that's replicated once. Oh, you can't see my cursor. Well, that doesn't make sense. Okay, so don't have a clicky pen. No. So the bottom block there would represent one each of those unique genotypes exactly once, and then again, and then again. So you've got 3 times each one of those genotypes in that field. In one of that plot there, you've got a number of plants from that genotype. They're all, let's say, homogeneous. They're all the same genotype because we've got those double haploids in there. And so plant breeding field trials have got a really long history in terms of the experimental design there. So you can do all this optimization within the field. You can place your genotypes in certain patterns so that, you know, you're capturing all the spatial variation, you've got blocking and all these types of things. And so you really have this formal sort of experiment that's going on. And then when I was sort of saying, oh, okay, we take those and we put those in different fields, different environments within the target population. On the right-hand side, I've just got a map of the US and just sort of example of where they might place their trial sites on that map there. So you've got a large number over on the East Coast. You've got a few down the middle there. That's where the Mississippi River is. And then on the west in Texas. And by placing those trials within those different locations, they're gauging what type of environmental conditions they've got there, but also what varieties or genotypes are well performing within those different environments. And so the one question that really is raised is, okay, so what's the target population of environments for this particular breeder? And it might be just one grouping on the right-hand side. It might be the Mississippi, or it might be Texas, or it might be some combination of them, or it might be subsets of them as well, depending on what they're targeting for production. And then another question is raised, well, let's say they were looking for something that was broadly adapted across multiple regions here. You know, why would they expect the performance in Texas, for instance, to be similar to the performance in the Mississippi? They're completely different environments. You're going to have different material that does different, performs differently in those different groups. And that's where this G by E comes about. And so G by E is essentially the relative response of different individuals to different environments. And so you may have some of those genotypes that do particularly well in Texas, but when they're placed in Mississippi, they don't perform as well, maybe in terms of yield production. And so you might have this re-ranking of the orders of the genotypes when they're placed in those different environments. And so if you're looking for a genotype that does particularly well in, say, Texas, you want it to be up the top. But then if it's placed in somewhere like Mississippi, the Mississippi River, where it's not well adapted, it's not going to be up the top of your performers. And so how do you make the selection if you want something that is sort of broadly adapted across both of those different regions? And that's what I was sort of saying, is that they want something that is broadly adapted within their target population, but they may also have different target populations of environments. And so how can they be placing the different genotypes in the different environments to sort of hit them? And then at the bottom, I'm sort of saying, oh, it's not just every year that this happens, that they're placing these trials from year to year. And as they're doing that, they're making that selections and they're going down the pipeline, and eventually they want to release something to farmers that's well performing and stable. Because you don't want to be releasing something to a farmer and all of a sudden we get a bad year, say, and therefore the production is going to be completely diminished. So this comes to a different ways that we can view G by E interaction. And on the plots here, I've got 2 examples of one is non-crossover and the other one is crossover. So in terms of non-crossover G by E, we can see that we've got 2 genotypes, G1 and G2. This is just a toy example, obviously. And we've got 2 environments, E1 and E2. And the classical form of non-crossover G by E is that the performance lines there, they never intersect. So that in E1, genotype G1 is the better performing line in terms of yield, say, and in E2, G1 is again the better performing line in E2. And so there's just a separation in the performance, but the rankings don't differ. So if you were to observe this type of G by E in your data, it's not going to affect your rankings between the two environments. So you know that if you select for G1, it is the better genotype in both those environments, regardless. There's a perfect correlation between the two environments there of 1. So that's another way of thinking. The correlation there is 1. What's just changing is the variance of the response. But of course, that's not always looks like that. It's not that simple. And that's where crossover G by E comes into place. And now we have a lack of correlation, or actually in this case, a perfect negative correlation, a correlation of -1. But I'm sort of saying now that the variation doesn't change in the two environments. It's just a complete reordering of the rankings there. So that if you were to select for environment 2, you'd want to be selecting G1. But if you're selecting for environment 1, you want to be selecting G2. And if those ones are representative of the target population, you want to be essentially selecting the right or specifically adapted genotype for those two environments. And another way of thinking about this is that if we were to look at the X's here, they would represent, say, the average breeding value across the two environments. And we can see that the average for G1 is higher than G2. And that it means that it's consistently higher for both those environments. Whereas in terms of the crossover case, we can see the average breeding value actually reverts to 0 for both of them. So there's no one good genotype that does well over both of them in this case. You need to be selecting for the specifically adapted genotypes for the two environments. Oop. And of course, it's not just as sort of simple as that. We don't just have these two specific cases where it's either this non-crossover form or this crossover form. What you end up having is actually some combination of both. And that's what's sort of happening again in this example here. We're seeing that the dotted line represents sort of the environment mean yield, say. So it's just the mean of the two environments. We can see that environment 2, the mean is higher than environment 1. So that's what's happening there. So we've got that E component. We can also see that on average, G1 is better than G2. So we've got a G component, and there's a re-ranking and everything, and a separation of variances. So we've got a G by E. So the question is that the two non-crossover and crossover, okay, they're well and good when they're separate, but how do you take something that looks like this and actually pull those bits out of it in order to select for, say, your overall performance across your environments? And how do you select for things like resilience or robustness or stability, or however you want to call it, in terms of the response to different environments from something that looks like that? Because if you were to come along and just select on the average, you'd select G1 because it has an average breeding value which is higher than G2. But if you were to place that genotype in an environment that looks like E1, that's where that reduction in yield comes from. And so what you really want to be doing is making sure that, okay, if you're selecting for averages, you know what you're selecting over. You don't want to be selecting over those re-rankings. You want to be selecting over the components that's causing just the separation. And of course, as a breeder, what you would actually like to have is, say, G3, which is broadly adapted to both of those environments. The average breeding value for G3 now is a little bit higher than G1, but actually in both of the environments, it does much better than the others, or sorry, a little bit less than the others, but we're still stable across those two environments. So G3 would be that broadly adapted genotype that you'd like. And in the example, if E1 represented, say, Texas, and E2 represented, say, Mississippi, G3 is that genotype that you could place in both of those types of environments, and it's going to perform well, consistently well. But again, it's all well and good with these little sort of examples, but what happens when we start having, a lot more genotypes to select from? I sort of said at the top there, you've got maybe, 10,000 individuals you've got to select for. And looking at these little regression lines, it becomes cumbersome, obviously. So you need some sort of a metric that you can summarize your performance or your average performance and summarize your responsiveness or stability as well. And it just becomes then a selection index essentially on those two components. So this is just sort of showing what we might like to construct is maybe on the y-axis, it looks like a performance measure. So you want something that's high performing on average. You want something that's up there where G3 is. And on the y-axis, sorry, x-axis, I've got those modeled up. On the x-axis, we're looking at, say, the stability or the responsive, however you want to define that. And so that what you'd like is something that is more stable. So you want something at the top left up there. And that would be that G3 in the example before. What we're seeing here with G1 and G2 is that, yes, G1 is on average higher than G2, but they've got about the same stability rating because they were both quite responsive to the other environment that they weren't well adapted to. So that's what you sort of want to get at. And within sort of the plant breeding, I guess, realm or is that these factor analytic selection tools have done a pretty good job of forming this selection index. I just want to talk about sort of what that is and what they're doing essentially. So they provide measures of performance or average performance, as well as a measure of stability for each one of those genotypes. So you're going to get out that little plot, the little 2D biplot for performance and stability. But they're based on the fact that these types of models can disentangle, pull apart the non-crossover from the crossover. So that's what they're built on, so that they could have an average over that non-crossover component and have a measure of stability of that re-rankings. That's what they're sort of the core of what they want to be able to do. And they come from fitting a linear mix model, essentially. I'll just show what that looks like. So we might have all of those different environments, say all of Texas and the Mississippi and across in the East Coast. We have phenotypes. We've got, I said before, sort of maybe 3 blocks or 3 replicates of each one of those genotypes. So you get a phenotype for each genotype in each block within each environment and then across everything. So you get, a large amount of data there, and in the Y, you might be fitting your fixed effects. It might be environment means or whatever it is. It's not particularly important to what's going to happen next. On the back end, we fit some non-genetic effects, some random effects there in the B, and some residuals, that everything that's left over. Within plant breeding, there's lots of spatial variation that occurs within the field, so you're accounting for all of that within the model. But the point really is that you just sort of fit some sort of a model to your data, and the crux of what this is all coming out of is the U part. So the U represents what we say random G by E effects, which gives you a breeding value or an estimated breeding value for each genotype by environment combination. So if you have a large number of genotypes and a large number of environments, this vector or these predictions, these estimated effects becomes very big. And the whole point of what I was saying before about these summaries was because if you were to have, let's say, 10,000 genotypes and 100 environments, you're already getting at 1 million combinations. And that just keeps growing bigger. So when you're making a selection decision, how are you going to select with having essentially 100 or 1,000 data points for each genotype? It becomes very difficult. So those summaries or those 2D plots and selection index reduces the dimension of that problem. And so what we want to take essentially is something that is very large in number of breeding values and reduce the rank of that so that we can make those selections for performance and stability. And what we fit to this particular vector is a factor analytic model, and that's a special type of random regression. So I'll just go through what that sort of looks like. So if we think about an ordinary random regression, and hopefully I've done my picture right, let's see. We've got our y-axis there down the middle, because I've assumed that I've got an environmental covariate that's been centered. So I've got minuses and positives, just so that what we then have is the intercept there on the y-axis, and that's going to represent the average performance of that genotype in an average environment. We've got a regression line that's going through our data points there. So each one of those black points there now represents the performance, the yield performance in those different environments. So for this little toy example, we've got 4 environments now. And what we're doing is we're fitting a regression line through that. The slope of that line represents the reaction of that genotype on that particular environmental covariate. And of course, you could have a whole raft of environmental covariates that are going into your model as well. This is just one, minimum humidity. You could also have temperature, whatever's driving the different reactions, responses in the different environments. And the point here is that if you were to make just an ordinary random regression, you know what the covariate is. You've measured the covariate in some manner on each one of your environments. So you have a large number of environments, but each one of those environments might have different covariates that are measured, and you have a gradient essentially that's going across those environments. And that's just the standard sort of random regression model. In terms of what a factor analytic model does is that says, well, let's just assume that we don't know what those covariates are. We've not measured rainfall, we've not measured temperature in each one of our environments. All we have, say, is the yield performance data. Apologies for a bit more of the algebra there, but on the left-hand side there, the random regression model is fitting a different intercept for each one of your genotypes. So if you have a large number of genotypes, you've got an average value for each one of them. You've got the known covariates there in the S. That's just saying that you're measuring different covariates, rainfall, temperature, whatever it is within this matrix, and you're getting a different reaction onto them, the slopes, onto each one of those covariates. On top of that, you're also modeling the little deviations for the environment-specific parts as well. So that's just the normal random regression or the reaction norm that happens. On top of that, with the factor analytic, just sort of says, well, we don't know those covariates anymore, so we're going to estimate those from the data. And those are what's coming now in the lambda component. Everything else looks similar. We don't have fit explicit intercepts because it gets captured within that latent environmental covariates. But another way of just viewing this is that the part on the left there is the covariates are known, but the part on the right is acting like a principal component analysis, where we're actually now estimating principal components of our data. And instead of having known bits on measured, on temperature or whatever it is, we now have things that are estimated from the phenotypic data. And so the factor analytics type model or regression looks very similar here on the right-hand side to before. It's just that now we don't have like what was before minimum humidity. It's just some latent variable that's been estimated. And that's coming directly from the phenotypes themselves. And I'll explain a little bit what that means with the different values in a sec. But what we'd like to get out of this, because we don't fit an intercept, is some value of average performance. And so we say, okay, we can take the mean value of the latent environmental covariate there. We can take the mean value on the regression line, and that gives us a measure of overall performance for that particular genotype. And that's what's going there with G4. And I sort of say, well, we might have another one down there in G5, and that's a bit lower. But the idea there is this is similar to before, is that you're just getting measures of average performance over these different environments by taking the value of the regression line at the mean. And the conjecture is that because those regression lines don't intersect, you're still averaging over non-crossover variation. So you're still averaging over the bits that you'd like to, because it's not included in this particular regression. But if we were to fit more latent covariates, they're capturing the re-rankings in those components. And so that's been coming out into the deviations there from the regression line. So another way of thinking about it is the regression line is representing your average performance for that line, and now the deviations around that is representing the re-rankings that's occurring. And so that's what I'm sort of saying in this part here, that the first part is just taking the average value of that regression line for giving some measure of overall performance. And the next part is looking at the deviations or the root mean square, the variation around that line. And so if you're looking for a selection index, what you'd like is about a high value of overall performance, so a high slope, because that's saying that on average it's quite high. And you'd like the deviations to be low around that, because therefore it's always at the expected value, which is quite high across them. How am I doing for time? So what I'll do is I'll show an example of how you fit this to some data and see what that actually looks like in real data. Hopefully that sort of makes a bit, but actually I'm not going to use real data. I'm just going to quickly simulate some. And with colleagues in the Highlander lab, we developed a framework to actually simulate G by E, and we put that within our R package FieldSimR, which is available on CRAN. But the idea here is that we're able to simulate it for a large number of environments, large number of genotypes. That's pretty realistic. Well, I mean, it's at the end of the day, it's a statistical model, but I mean, it's kind of matches what we see in real data. And we're able to actually construct data sets from that simulated G by E. And one of the key components for this is, I won't go through the math, it's not really that important, but one of the key components is that we might construct correlations between environments. And so that's what's happening here on the right-hand side, is that this heat map here, each one of these little squares represents a pairwise correlation between two environments. And so I'm showing here, I think about 200 environments, just as an example. So if we see the reds here, if there's more red, it means they're more correlated. And that's the first example up the top. And if this goes down to yellow and blue, it's negatively correlated. So there's a lot more of what we say re-ranking G by E, because if you've got two environments which have a correlation of 1, there's no re-rankings, as I was saying before. But if you have a correlation of, say, -1, you've got perfect re-rankings, reshuffling of them. So what we're doing here is just simulating some correlation matrices, and we can see the distribution of those pairwise correlations at the top. And we say that, okay, we've got about a mean correlation of 0.6. So on average, between these 200 environments, you've got a mean correlation of 0.6 with some variation around that. We can see that that's what's happening with the distribution. And then the one below, we've got a lot more of that re-ranking going on. We can see the distributions going all the way back to -1. So it's capturing a lot more of that re-ranking. And so what I'm just going to do is classify the top plot as low G by E, because there's much less re-rankings within that. So the best performing genotypes in some of those environments, or most of those environments, are the same. Whereas in the bottom one, I'm going to say that's high G by E in terms of the re-rankings, because there's a lot more of that switching going on. So if, say, a plant breeder or an animal breeder, I mean, animal breeder might not have 200 environments, but if they had the top set of environments as their TPE, it's a lot easier to select for that one genotype that does well across all of them, because a lot of those environments they're quite similar in terms of the performance that they're being observed. In the extreme, if you had a correlation of 1 across all your environments, again, you just have to select one genotype and it's going to do well or the best over all of them. When you get down to the high G by E scenario, finding that one genotype becomes very difficult, because all of those environments are so different. And all those, the performances are so much different as well. Finding that one that does well is the needle in the haystack that the plant breeder or the animal breeder or whatever it is wants to find. So we just simulated a little bit of data there. We then went and fit some of these models to this data, and that's what we're getting here. And so on the left-hand side here, we're getting these principal, let's just call them principal components, because it's easy to sort of think about like that. And each one of these dot points represents an environment. In this case, I've reduced it just down to 20, just to sort of as a demonstration. And we've got 2 genotypes here as a comparison. So if we just look completely at the first plot, the far left, we've got the regression lines for the 2 genotypes. I think it's ID 73 and ID 179. They're simulated. And we can see that the regression lines there, they don't intersect. They just spread out. So what's happening here is that this is representative of that non-crossover G by E. On the y-axis, we've got the predicted breeding values for each one of those environments for those two genotypes. And so the ID73, which is in red, has got a much higher slope for that principal component. So it's got a much higher average breeding value. Whereas ID179 has got that negative slope there. So again, if you want to select for that average performing or the highest average performing line across those environments, it would be definitely ID73. And this with the low G by E scenario. In terms of the other principal components in this model, there's a little bit of variation on the second one and then a little bit more on the third one there. But the main point there is that you can see the intersecting of those lines. So those principal components are capturing the crossing over or the crossover G by E. Those are the ones that are capturing the re-rankings between the environments. And they're the ones that are causing the deviations from the first principal component regression line. So in the extreme, if you had a slope of 0 for the second and the third principal component, your dot points on the first plot would be all sitting on the line because you're not getting any more contributions from the rest. It's stable for those two different principal components. So have a high average performance. And so it has a high response to the non-crossover G by E. And then it's stable to the crossover G by E where all the re-rankings is. That would be the sort of the ideal genotype. And so on the right-hand side, there is what I've done is just summarized that average performance mark as well as the deviations, so the stability mark, and put it on that plot there. So we can clearly see that ID73 is up the top of, say, all of the 200 different genotypes that we've simulated. That's one of the, the winners. It's quite broadly adapted. Whereas ID179 is on average really poor, and it's got a bit more of that variation around its regression line. So it's got a bit more of instability there. So you want that top left point. But if we do the same to say fitting something with high G by E, and that was the other scenario that we simulated, we can see that the first principal component no longer captures any of that non-crossover. There's already re-rankings in here. And that makes sense because like I was saying, the average correlation in those particular environments was much lower. I think it was around about 2.2 or something. And so there's a lot more re-ranking within that data that we simulated. So it's not unexpected that when you're pulling that variation out with these models is that you're coming into that first principal component. And so what this means as well is that it no longer captures an average performance over non-crossover G by E because it's clearly crossing over there. And so you're not making the averages that you would think that you are. In fact, if we look at all of those principal components, they're all capturing some form of crossover interaction. So it's fair to say that the predominant variation in this particular data is crossover variation or re-rankings between the environments. And so the question raises, and this is, although it was a simulated data set, this occurs in real life as well. And the question is always raised, then how do we select for a broadly adapted genotype when we've got this type of or level of G by E? And how do we actually explicitly disentangle those components so that we can still select for the average performance that we'd like to? Is it possible? And the question, the answer really is yes. And I'm just going to flick through a couple of these just because of time. But that really raised the limitations of these particular selection tools was that, you're relying on this principal component rotation to rotate non-crossover variation into the first principal component. And mostly it sort of occurs as a natural artifact of plant breeding data because there's a lot of that separation out within plant breeding data, but it doesn't actually guarantee it. And then on top of that, okay, if we're using some principal component rotation, why not use a different one, and et cetera, et cetera. But what happens when you do actually capture the re-rankings in that first principal component? how do you move forward with that? And traditionally what was occurred was that if we look at the environments there in the first principal component, you'd say, well, maybe there's two different environment groups, because on the left-hand side, they might be more similar than on the right-hand side of that intersection there. And you would look at your data and say, well, is there natural groups of environments there that we could actually separate out and select for differently? And most often it's no, or it doesn't represent something that's particularly meaningful. And the other option was, okay, what happens if you just remove those from the left-hand side from the data and don't actually include them within your selections? But there is actually still a way to still separate out those different components. And that was really sort of a motivation to some of my work, is that, okay, how do we look at the mean breeding value within the data and look at the variation associated with that mean and move that to the non-crossover component? And so what really is left over is the true deviations, is the re-rankings, is the crossover. And what it requires is us to redefine essentially what non-crossover is, or explicitly define what crossover G by E is. And that is all variation attributed to perfect positive correlation with the average breeding values. And it's a little bit long-winded, but the point is that if we were to set up the average breeding values across, again, this little dummy example, and say that all the variation that is perfectly positively correlated with it, we want to capture that somewhere. And so in this example here, those regression lines are perfectly I knew this would happen. Are perfectly positive correlated with those average breeding values, because they're just spreading out on the ends or shrinking. They're never re-ranking. So if you were to take a correlation between those average breeding values there on the X's and the slopes of those two regression lines, the correlation is still 1. And so you can do that for anything that's doing this, provided that they're going through the same rankings as the intercepts, as the average breeding values would. And I'm not going to go, again, too much through the algebra, but firstly, you would need to construct those average breeding values. And then we look at the variation associated with those breeding values. And we essentially move those from the full set of breeding values to just the average. And I'll probably put a little bit too much here, but what I'll just show you is what happens. Skip through that. And so in practice, on the left-hand side, there was the PCA approach. And we saw that the first principal component captured that crossover. There was crossover G by E going on in there. And we said, well, we can't take our averages over here. And with the new approach, what it does is that it actually causes that rotation to create this non-crossover here. So you can see that now we've got the fan. And so that first principal component, which is no longer actually a principal component, the first factor, explicitly captures non-crossover G by E, because we can see there's no intersection there. And you say, well, why is this important? And it's important because I still want to be able to select on that average breeding value and not capture any of the crossover, because in the remaining part, when we're selecting for stability and responsiveness, that's where we want to be capturing the crossover. So we can construct, and now we can construct our overall performance that we want there. And we can now construct our selection index on the right-hand side, which is overall performance versus stability. And if we were to compare the two from the two methods, we now have really vastly different plots. And that's because, again, the first one on the left was never actually capturing the true average performances over. It was all based on the wrong rotation, essentially. And we can see now what we want to do is we've actually identified two genotypes up there in the top left, which would be those high-performing stable lines across all of those sets of environments. So that's really what the crux is all about, is that how do we actually construct the right index? And those two there were not the same two over there on the top left either. So they've now actually come out. And again, you sort of say, well, why is this all important? And this was the plant breeding case, but if I think about sort of an equivalent animal breeding setting, it would be like having a random regression on some sort of trait, let's say it's temperature, some sort of temperature gradient, and you would have fit a regression on that, and you've got intercepts and you've got slopes. If you've got a lot of covariance or correlation between your intercepts and your slopes, when you select on the response in terms of the slope, you're not just selecting on its resilience or its responsiveness, you're also selecting or indirectly selecting on still the mean performance, the intercept. So you're not actually, you're not maximizing your response to selection for that breeding objective. And so it's really important that you actually remove that variation first and then select on the slopes that have been adjusted for the correlation with the intercept. So that would be sort of an equivalent animal breeding sort of example. And then I've just sort of got here to just sort of say, well, if it just comes down to a selection problem, let's say we were to select on the old measures and say take the top 20 genotypes, which are based on the true there, and they're in the red, and you're having going to select a different top 20 based on the two different measures in terms of the old PCA type approach. And really the big difference doesn't come into the first sort of overall performance. What it comes into is when you're selecting for stability there on the right-hand side, and that's because the old PCA approach doesn't take enough of that variation associated with the mean out of the stability measure, and you're getting vastly different. And the practical application there is that you may think that you're selecting on something that is broadly adapted, but actually it's just because it's, high performing or the vice versa. You might be selecting on something that's just got associated still with its average performance and not its response to particular environments. And I just finish up by saying that the new approach really does separate out those components explicitly. It's not just, I've sort of mentioned, the plant breeding setting, but it's also like just quangen in general about taking something that is a G by E component and actually separating that out into components that are just associated with its mean and the variance that is causing re-rankings. What we're also getting here is actually two new traits that are independent, so you can select for average performance and stability within a selection index, and there's no correlation anymore between those two components. And again, I sort of mentioned the last point there with the animal breeding setting about taking away the variation associated with the intercept or the average breeding value before selecting on its resilience, so that you're actually selecting on the right thing. It does raise a question though about, at what point does this selection for broad adaptation across all these environments just become a bit meaningless? But provided you've got some variation associated with the mean, you can still make progress. It just depends on what level that is. And the other side of the coin is that if you are able to actually group environments into meaningful cohorts, then that would make, you've got more correlation within each of those groups, makes a bit more sense. It just becomes difficult when you can't do that in a meaningful way from year to year, that they don't keep showing up, that those groups of environments represent the same things. And we're looking at putting all this stuff sort of within simulation to say, okay, that's all well and good, but what's actually my effect on genetic gain over time? And the other side of the coin too is that you can do all this within GWAS or a genome-wide association studies, because you're no longer just looking for markers that are contributing to the average breeding value, but also which ones are contributing to responsiveness and all the rest of it, and which ones are contributing to the non-crossing over, or which ones are really affecting the re-rankings. And that's what this is really about. Which ones are actually affecting the re-rankings, not just variation with the mean. But I think that's all I've got. Yeah. So with that, I'd like to say thank you, and happy to take any questions.
So let's suppose you now have variables. You're observing them.
Yep.
So one thing is maybe it will help you to give an interpretation to each one of the factors. But it's temperature, it's something else that's causing change, it's ranking. Like, I guess this is one idea, but what else, right, could you use for, you know, these variables for in terms of factor analysis?
So-
Can you just repeat the question for the?
Yeah. So the question was that, let's say you now have the environmental variables. So let's say you've got the rainfall and you've got the temperature measured. One thing that you can do then is try and ascribe meaning to these particular factors. You know, so one way that they typically already do is do some sort of a correlation between the maximum temperature or whatever it is and these particular factors to try and say, okay, that principal component is being driven by this sort of thing. And they've been sort of doing that for a little while. Problem within plant breeding is that generally each one of these principal components is not driven by one particular covariate or variable. It's some raft or combination of a number of ones. But that's definitely something that you can do.
But this is also what principal component does, right? It's going to be a combination.
By definition, exactly. By definition, it is some complex combination of these things that go into it. But in terms of this particular research, you can say that, well, I'm not really interested in what variables are driving the mean per se, maybe. But once I take the variation with the mean away, which ones are actually really driving the re-rankings that I see? So it's directly applicable to this as well. And also on top of that, it's like I was sort of saying, is that if you actually fit those variables in the first place, then you can see explicitly as well which ones are driving the, you know, the re-rankings by doing those adjustments that you need to do.
I'm from animal breeding background, so the question I don't think makes sense. But I wanted to ask, I understand the importance of keeping a consistency and stability of the product across years, but when you look at different environments alone, if the experimental design of the different plots allows us to calculate progress, and if the target population environment is already kind of a thing, with already traits, and you can go in the index and you can get those random effects of breeding values as a G by 800, then why are we interested into having this robustness across environments rather than targeting and optimizing and creating optimized tailored products for specific breeding locations, specific locations of production, in that case, and risk losing some of the variability that would come from those individuals that would perform best and optimally in a specific environment, but not in one.
I'm going to have to try and repeat the question, but this is my summary of the question. Because we've essentially got everything tested everywhere, that why aren't we sort of trying to target specific environments for our products rather than going for broad adaptation and stability across all environments? Because it makes sense that you would lose a bit of variation, but you're actually gaining efficiency because you're going to choose the best things in smaller number, but that are more correlated. Okay. The short answer is that's another one of the breeding goals as well, is that they do try and do that. Making things into smaller groups that are more correlated, more similar, you know, and then targeting that, they are trying to do that. Problem is that if you can't do that, it's always easier to sort of group things into smaller regions in terms of geography, because it makes sense that that's the market. Those are the farmers that are going to buy the seed for that market. They're in this area. You know, it's like in the example, you know, target Texas, and they do that, and target Mississippi differently. And the question really then comes into, when I'm talking about broad adaptation, is that, well, maybe there is room to move material from one area to the other, you know, still targeting those separately, but there might be something over here that does particularly well over here in that case. And I didn't really show in this talk, but for those sets of environments in the USA, applying these methods to that and mapping out what the environments look like the particular breeding program didn't really know that the ones on the Atlantic and in the Mississippi were actually quite well correlated. So they could start targeting and moving material between those two different regions. Previously, what they did was kept everything separate. Same with Texas. Like, I mean, they knew that the Texas was always going to be very different. It's completely different environments over there. But you only need one or two genotypes to actually make a good influence. But it's about having the data and moving it and moving the material over. But yeah, they definitely do that sort of thing, like you're saying, and some combination of all. So this was all captured in the PhD, and I guess one of the benefits of the way I did my career and starting out with doing a bit of work in Australia before doing the PhD was that I really brought my research from Australia. That this was it. So my fellowship, to answer your question, is a continuation of the research that I started back in Australia and this type of thing. There's lots in this sort of space. I mean, this is very plant breeding heavy so far, but I want to be here at Roslyn applying it more to animal settings as well.
So have you applied this method to real data already? And are there specific systems or species that would probably, like, the industry would be particularly interested in this? And like, do you have to think of any special considerations?
Yeah, I mean, so it has been applied to real data already. I guess the way that these types of models are construed is that they are widely applicable. And so there's not really any, so far has been, oh, we need to consider this in this way. It's mostly about, okay, in these models that don't have known covariates, that so everything's latent, is what does it actually mean that's driving it? That's usually the big question about that. But it's going to be applied to a few more data, like real data sets and everything in as well. Over one point I didn't put up here is we're actually hiring a postdoc to work in this space. So yeah, it's to apply it to more stuff. So, yeah.
Any other questions? Then thank you again, and thank you everyone for joining us in person and online. And yeah.